Well, thanks everybody for attending tonight's third session of our chronic wasting disease webinar, brought to you by the University of Minnesota Center for Prion Research and Outreach, otherwise known as MinPro. I'm Mark Schwabenlander, and I'm the CWD Research and Outreach Manager for MinPro, and I'll be moderating this webinar and the webinar next week, our final and fourth session. A few quick webinar housekeeping items. First of all, this session is recorded and it will subsequently be available on our MinPro website, minpro.umn.edu/outreach. For the attendees, the cameras and microphones will be off during the session. If you do have questions, please enter them into the Q&A tab. If you are new to Zoom, if you follow along, I can show you how to enter a question in Q&A. There is, at the bottom of the Zoom screen, there's a Q&A tab to click on. If you do not see that, you can hover your mouse over the bottom area of the screen and it should appear. Next, if you click in the text box, you can type your question. And if you want to send that question anonymously, click the box next to send anonymously, and we won't know who sent that question, but we will answer it equally. And finally, you click that send button and it will come through. You can ask your questions anytime during the session, and we will answer those at the end after Corey is done presenting tonight. If you're listening via telephone or watching the recording, please send any questions about tonight's seminar to our MinPro email, minpro@umn.edu. So without further ado, tonight's presentation will be led by Corey Anderson. Corey is currently part of the Center for Infectious Disease Research and Policy, or CIDRAP. He's part of their CWD program. He joined CIDRAP as a graduate research assistant in January 2019 after earning his Master of Public Health degree from the University of Minnesota. He is currently pursuing a doctorate degree in environmental health at the University of Minnesota with a concentration in infectious diseases. So with that, I will unshare my screen and let Corey share his, and he can take it away.
Perfect. Well, thank you, Mark, and thank you to the rest of the MinPro team. We really enjoy working with you. It's been a fun partnership, and I'm excited to see where we go from here. So like Mark said, I'm Corey Anderson. I'm a research assistant with the Center for Infectious Disease Research and Policy. And today I'll be talking about the human factor of chronic wasting disease. Let's see if I can get this to advance. Perfect. So I'm assuming a lot of you were in attendance for the last couple of presentations where Peter did an excellent review of CWD and the ecology. So I won't spend a whole lot of time on this. If it gets confusing, you weren't there for the previous ones, I'd recommend going back and watching the recordings. But just some basic background here before we start. So with chronic wasting disease, we know that it's a prion disease in cervids, which includes, deer, elk, moose, and caribou. It's always fatal to infected cervids. Unfortunately, there's no complete genetic resistance to CWD that is known. There are no vaccines or effective therapeutics. And I really want to emphasize this from the beginning. There is no current evidence that CWD has infected humans to this point. When it comes to an animal that's infected with CWD, like other prion diseases, CWD has a notoriously long incubation period. So on average, it's anywhere from 18 to 24 months. During that incubation period, an infected animal sheds notoriously resilient infectious prions for most of that incubation period, even while it appears totally healthy. So these prions are shed in the saliva, the feces, urine, blood, antler velvet, and semen. After that 18 to 24 month incubation period, there's a short window of time where the animal shows clinical symptoms and then eventually succumbs to the disease. During that short window of time, those symptoms could include weight loss or wasting, lack of coordination, altered gaits, excessive salivation, lack of fear of humans. I'm sure a lot of you have seen this map before. Thank you to Brian Richards for putting this together. Brian's with USGS. This is just the North American distribution of CWD. But as far as worldwide picture, CWD has so far been found in 26 US states, 3 Canadian provinces, South Korea, Finland, Norway, and Sweden. When it comes to Minnesota, again, a lot of you are probably familiar with this, assuming you're tuning in from Minnesota. But you can see we've had a number of interactions with CWD detections, whether that's on cervid farms or in wild populations. Of course, in the Southeast region, that's where we really have this sort of endemic area where CWD has been found year after year since 2016, when 3 wild deer were detected there. And then I just want to highlight a couple of other kind of current detections that are notable. So you'll see number 13, that's up in Crow Wing County. They recently detected a wild deer there in 2019, which was actually detected because of surveillance initiated from a detection in a farm deer a couple of years prior to that. So far, it's just been the 1 positive wild deer there. And then number 16 is a recent detection in 2020 that's in Dakota County in the South Metro area, where actually a clinically ill deer was spotted by a resident. I believe DNR was called. The animal ended up testing positive for CWD, and so they're hoping to do more surveillance there. And of course, next week's presentation is actually being led by Minnesota DNR staff, so I'm sure they could tell you a lot more about that. And then actually just a final thing to highlight, number 17 happened just a week or so ago. There was a recent detection in a Houston County deer farm. And so that's something to keep tabs on as well. So when it comes to CWD and human health, there are some things we know, and then of course there's always things that we don't know. So what do we know at this point? Well, I think you've probably heard from Peter and what I just explained. CWD continues to spread in both wild and farm cervids. With that spread, we know that these infected animals are shedding infected prions or infectious prions onto the landscape. And again, those prions can remain on the landscape for years to potentially decades. And so as the disease continues to transmit, we also have growing environmental contamination. We know that unique CWD prion strains exist, and I'll cover this a little more later in the presentation. And also, we know that human exposure to CWD is increasing as the disease increases in animal populations. There are some specific estimates, they're a little outdated, but it's safe to say that venison from thousands of positive cervids is consumed each year. I would even venture to say tens of thousands of positive cervids, around 20,000 is probably a safe guess. But of course, the million-dollar question and what remains unknown is whether CWD will cross the species barrier and cause disease in humans. That's what a lot of researchers are focusing on. Of course, a lot of media attention is spent on this issue. And so I'll just kind of scratch the surface and try to clarify some things, or at least explain some of the science behind this issue. So of course, when you have a question that's unanswered, it's always helpful to look back in the past on some lessons learned. And so we really have kind of 2 different stories when it comes to human risk as far as prion diseases go from another animal. So this first prion disease you've likely heard about, it's called scrapie. It is a prion disease in sheep and goats that's actually been described for hundreds of years. I think the first written description of it was in 1732 or 1730s. And so obviously it's been around and been known for quite some time. Scrapie is similar to CWD in that the infectious prions are shed in bodily fluids. I think the highest concentration of scrapie prions are found in the afterbirth. But fortunately, since scrapie basically it infects domesticated animals, it's basically capable of being controlled much easier than CWD, which is in a wild population for the most part. And so national efforts have largely controlled this disease over the years. Just as an example, the USDA has a national scrapie eradication program, which has reduced this disease in goats and sheep dramatically. An important note, and the reason why this is a lesson, is even though it's been around for hundreds of years and known for hundreds of years, there has not been a single case of human prion disease that's been derived from scrapie or been caused by scrapie. On the other hand, we have bovine spongiform encephalopathy, or BSE, mad cow disease as it's referred to by some. And this is a prion disease in cattle. It was actually first reported in 1986, and a vast majority of the cases occurred in the United Kingdom. The origin of BSE how it got into cattle. There's some speculation. We won't really get into that. It's not confirmed, but essentially what happened is BSE got into cattle. These cattle were slaughtered, of course, and instead of just throwing away the waste, that waste was rendered down and actually fed back to calves as a source of protein. And so that meat and bone meal was fed back to calves. The BSE prions survived that rendering process. And essentially it was kind of this amplified transmission that occurred. Of course, that was detected when the problem was found, that feeding practice was identified as a risk factor. Restrictions were put in place and the cases of BSE dropped. Again, it helps to have these diseases in domesticated populations where you kind of have control over them. Unfortunately, before that restriction was put into place, an estimated 500,000 to 1 million BSE infected cattle entered the human food supply. That number is debated quite a bit. There's some people who think it's quite a bit more, but regardless, quite a few infected cattle entered. And so for 10 years, it was really debated as to what the risk was. There was kind of some different thoughts and opinions on that, but unfortunately, everyone's worst nightmare came true in 1996. So 10 years after that first detection of BSE in cattle, the first human case of variant Creutzfeldt-Jakob disease was identified. As of 2018, at least 231 cases of VCJD have been detected in 12 different countries. A vast majority of those cases are linked to consumption of beef contaminated with those BSE prions, although a few of them were caused by blood transfusions. That risk, again, has been basically mitigated through donor deferral. With VCGAD, again, it follows kind of that notorious TSE or prion disease pattern with a decade-long incubation period on average. The median age at death is 28 years old, which is much different than sporadic CJD, or the most common human prion disease form, which most commonly affects older individuals. And then interestingly enough, studies in the UK have actually identified thousands of subclinical carriers. And it's really unknown what the implications of carriers are. These studies were done looking at appendix samples of UK residents. And some people believe that up to 1 in 2,000 UK residents might be, or basically they found abnormal prion deposits in their appendix, and they're really not sure what that means at this time. So again, something to keep tabs on moving forward. And so, like I said, we kind of have these two diseases, these two previous known TSEs with kind of different directions as far as human risk goes. And so a lot of people like to compare the two and look at CWD, and it's really a picture of where does CWD stand? Which TSE is it more similar to as far as human risk goes? And that's what I'll try to explain. Just a spoiler, I'm not going to have any definitive answer on this, but I'll try to explain the science that's gone into looking at the species barrier between cervids and humans with CWD. And so I just want to take a step back, and this is kind of a high level understanding, just a very vague understanding of the prion disease species barrier. I just want everyone to know that when it comes to prion diseases and their species barrier, it's important to know that it's a direct molecular interaction that's taking place between the normal prion protein of the exposed individual, whether that's a person or an animal, and the disease-causing prion. So in this case, it would be essentially a human normal prion protein, and the disease-causing prion would be CWD. And so I liken it to a lock and key, where a human could be exposed to a certain type of prion, and just like with a car, you could use the wrong key, put it into a car, of course, turn it, and nothing would happen. So no disease would be caused. That process would not begin. However, unfortunately, in some instances, that molecular interaction takes place and a chain reaction occurs. So in that case, the key would be correct. It would be a right fit for the car. The car would turn over and that process begins. So that's really the disease process. And of course, it's much more complex than that. There's a lot of remaining considerations that have to be accounted for. So that includes genetics, dosages, the route of exposure, different tissue types, and beyond. So of course, I'm not going to be able to cover all of this today, and I don't even know myself what that really involves at a high level understanding. Another kind of unique feature of CWD compared to BSE that I thought is worth highlighting, they really have kind of two very different distributions as far as the prions go in the body. So in an infected bovine, the vast majority of the prions would be located in the central nervous system tissues of that animal, i.e. the brain and the spinal cord. With CWD, on the other hand, an infected animal would have these prions present in many of the tissues of that infected cervid, and that includes early and widespread accumulation in lymphoid tissues. That's actually presumed to be the reason why CWD is contagious, because it reaches the lymphoid tissues and peripheral tissues and would be capable of basically entering bodily fluids. On the other hand, with BSE, they were not shedding infectious prions in their saliva or in their feces or in their urine like deer were. Instead, it's important to remember that that infection only took off in that population because of humans basically feeding slaughtered cattle back to cattle. So it was almost kind of forced cannibalism in a way. But yeah, just an important kind of note there. And so when I talk about prions with CWD being widespread in the body, I just wanted to, cover some published papers here. So of course, this one detected prions in the saliva and the blood, which we know about. It's been found in the fat of deer with CWD, the salivary, urinary, and intestinal tissues. But then kind of most important as far as the human risk goes is that CWD prions have been found in the skeletal muscle of deer with CWD. So again, a unique feature in that BSE would not necessarily be found in the skeletal muscle. The food that humans would have been eating would have had to have been contaminated with some of the central nervous system tissue, which actually did happen quite often because of processing activities and processing actions back then. Like I said, I'll touch on prion strains. This kind of gets beyond my area of expertise and gets almost into the biochemical area. But like I said, I know enough to say that unique strains of CWD prions do exist. These prion strains are characterized by slight differences in their shape. And so you can see that very kind of elementary picture right there. It's simple, but it proves a good point where, these different infectious prions interact with that normal prion protein and then cause it to misfold into that certain shape. And so that's really what these strain variations are. These changes in shape can take place when the prion passes through new hosts. And so what that really means is that continued CWD transmission and persistence on the landscape will inevitably lead to more strains. And kind of the worrying part about different CWD strains or prion strains in general is that these strains can have unique host ranges. Basically, they are capable of infecting different species as far as between these strains go. And so this is just one paper kind of highlighting that point. And I'll just read the abstract here. This is a group out of Alberta that's been leading some important work in this area. But what the abstract says is that human and mouse prion proteins share a structural motif that regulates resistance to CWD or common CWD prion strains. Successful transmission of an emergent strain of CWD prion H95+ into mice resulted in infection. Thus, emergent CWD strains may have higher zoonotic potential than common strains. Again, I want to emphasize may have higher, it's not known, but again, it's something to keep tabs on and keep your eyes on as transmission in cervids continues. I just want to, as a disclaimer, say that if you have questions about strains, which I personally do as well, I would recommend checking out this webinar that my advisor, Mike Osterholm, hosted with Debbie McKenzie, who's actually part of that Alberta group. That's available on our CIDREP CWD YouTube channel. It's an hour long, but Debbie does an excellent job breaking down kind of the super sciencey part of prion strains and making it understandable. So if you have questions that I'm not able to answer, go and check that out. And so now I'll just kind of dive in and do, like I said, a surface level explanation or give some examples of the science that's been going on looking into the species barrier. And so when we look at the species barrier, there's really 3 different study types that have been used to assess what that barrier is and how robust it is between humans and cervids when it comes to CWD. So we have three study types. The first is observational or epidemiological studies. The next are in vitro studies, which are really just test tube experiments that take place outside of an organism. And then the third type are in vivo studies, or really animal model studies. Again, like I said, just scraping the surface, we actually have a lot more information on these studies and their results on our website. So I just picked some of the more interesting studies to talk about. When it comes to observational studies, this is always kind of an interesting one. So in 2005, there was a sportsman's feast that took place in upstate New York, and 81 individuals unintentionally consumed venison from a CWD positive deer. They did not know it was positive at the time, but of course afterwards they found out. And so because this was really a point source exposure where, you know, dozens of people were exposed to CWD, they thought it'd be a great study. And they actually asked each of these individuals to complete an exposure questionnaire, and they really have recurring health evaluations that take place. This paper actually was published in 2014, so a long while after that 2005 point source exposure. And to this day, there is still no evidence of prion infection among this group of individuals. And then another frequently cited paper was published in 2006. And this paper actually evaluated rates of human prion disease deaths in Colorado using death certificate data from 1979 to 2001. And they found no increase in the relative risk of sporadic CJD in residents of Colorado's CWD endemic counties, assuming that they might have had more exposure to CWD. They also noted that CJD rates did not increase over time. There are some people who use this to basically say, you know, this is evidence that CWD hasn't and will not infect humans, but it's important to note that this kind of happened early on. And so the authors, I just put one of their sentences here. The authors basically note that unknowns about the incubation period, the exposure, and clinical presentation, they basically said that this was sort of a baseline study. And so it'd be interesting to kind of do these on a decade interval basis, in my opinion. Of course, there are some major limitations when it comes to observational studies, especially with prion diseases. So these studies are really looking for a statistical significance connecting exposures and outcomes. That is really complicated by prion ecology just because of year to decade long incubation periods, very low attack rates. Remember with the BSE example, there's potentially millions of, or a million infected cattle that enter the human food supply and 231 total cases in humans. So a very low attack rate potentially. And then again, I think it's important to note that it's unknown how the clinical presentation in humans would look if infection did occur. A lot of people like to speculate and think it would be similar to VCJD, but it could be a more dementia-like picture. We don't know at this time. And that really makes it easy for connections to go unnoticed. Now, I'm not knocking on observational studies. I actually think they're entirely necessary and critical. Human prion disease surveillance in general is critically important moving forward. And it's important to keep having these studies on a recurring basis moving forward as exposure increases. Next, again, these in vitro studies, which are these test tube experiments. I'll just explain the science from a level that I can even understand. But basically what they're doing is exposing normal prion proteins of select species to various types of infectious prions. And so in that very elementary level image I put there, you could have normal human prion proteins, cervid prion proteins, whatever species, you name it, and you can pick the infectious prion, expose those normal prion proteins to that prion in a test tube and see whether conversion occurs. And then, of course, if conversion does occur or misfolding does occur, how efficient is that misfolding? And that really provides timely insight into the species barrier. Again, when you're working with animal models, which I'll get to in a bit, you're talking about years to decades of work. This has its limitations, of course, but it provides that kind of timely information that's needed. So I'm not going to cover any specific in vitro studies. There are a number of them that have involved CWD and normal human prion proteins. And actually most studies have shown CWD prions capable of converting normal human prion proteins. That's not anything to freak out about though, because that is not definite evidence that this will happen. The efficiency of conversion varies quite a bit between these studies. So as I wrote here, study conclusions range from severely limited efficiency, where they're basically saying that I doubt this would happen in real life, to some study or one study saying that the efficiency was more, or it was more efficient than BSE, which of course is a known pathogen to humans. So I think it's important to just note, I've talked to people who are actually experts in this area, and they really say that the results from these studies are highly dependent on the methods, materials, and tools used. That makes it really difficult to compare these results. And it's also really difficult to know which of these studies best represents a real world setting, or what study results we can really apply to a real world setting. So it offers really good insight, but it's really hard to understand human risk just based off of these studies alone. And then last, kind of this final component of studies that I'll briefly talk about. These are those in vivo studies, or these animal model studies. And some more common animal models that are used in CWD species barrier studies include transgenic mice, and then non-human primates. So squirrel monkeys and cynomolgus macaques are commonly used. And when it comes to routes of exposure, oftentimes these animals are intracerebrally inoculated with CWD prions. So that's basically injecting it straight into the brain. Obviously, that's not the most realistic route, but it's really just a proof of concept to see, if these animals are susceptible to CWD, or basically if they're not susceptible after being injected in the brain, you could kind of guess that they wouldn't be susceptible if they consumed prions orally. Of course, with that being said, they do still expose some of these animals through oral consumption. And then there's also some studies that look at skin scarification, just to basically replicate, if you were field dressing an animal and nicked your skin with a knife, whether that poses any risk. So first, with transgenic mice, again, I grouped these studies together just because a lot of them share similar results. And so I'll back up a bit and explain what transgenic mice are. These are also considered humanized mice, where it's basically they're designed in a lab or engineered in a lab that normal mouse prion protein is knocked out and actually replaced with a human prion protein gene, which in turn causes these mice to express normal human prion proteins. There's actually at least 6 labs conducting research using these mice. And actually, as far as I'm aware, there's no current evidence of prion disease found among humanized mice that are exposed to CWD, even following IC inoculation in these animals. And now just kind of moving into our non-human primates and what those results show. These studies involve squirrel monkeys. There's actually a few of them from 2 different groups. And all of their results indicate that squirrel monkeys are highly susceptible to CWD. In one of the studies, all of the animals were IC inoculated, so injected straight in the brain with CWD. In the NIH study, they actually fed the squirrel monkeys brain material with CWD prions in them. Regardless of route of exposure, all of the exposed squirrel monkeys were infected. Excuse me. But it's also important to note that previous studies have been done using other TSCs such as scrapie, and squirrel monkeys are actually susceptible to scrapie as well. And so, of course, backing up again, humans have basically been shown not to be at risk of becoming infected with a prion disease after exposure to scrapie. And so this really kind of brings into question how realistic of a model are these for humans and the risk that CWD poses to humans. And so one of the study authors basically said that caution is warranted when interpreting these results. And then now I'll just kind of briefly touch on a more controversial animal model, and this has been ongoing for a few years now, but cynomolgus macaques have been kind of subject to controversy when it comes to exposure to CWD prions. And so there have been really several studies that have looked at whether or not macaques can become infected after exposure. And we have basically 2 opposing conclusions from studies. The first results come out of NIH and Rocky Mountain Labs, where they exposed macaques via IC injection and then fed others brain material with CWD. And basically since 2009, they've been reporting that they have not found infections in any of those cynomolgus macaques. Of course, the media kind of took off in 2017 when this Canadian and German study, the researchers presented their preliminary results and basically said that they have the first evidence suggesting that prion infections took place in 4 cynomolgus macaques. More interestingly, and this is where the media kind of blew up with this, was that they reported that 2 of the infected macaques they had were actually fed skeletal muscle from an asymptomatic CWD positive white-tailed deer. Now, of course, when you're talking about accurate or realistic routes of exposure, that would be the route of exposure that is most applicable to humans. And so not only did they say they found infections in cynomolgus macaques, which had not been done before, they said that they were infected after eating the skeletal muscle. And so, of course, this took off quite a bit. At this point, I really want to emphasize that the Canadian and German study is still unpublished. It has not been peer reviewed. There's a lot of kind of debate going on as to why they got the results they did. Yeah, it'll just be interesting to see what comes of that moving forward. But again, that's kind of brought a lot more attention to this issue, and it remains to be seen what those results are and what they mean. Of course, with that being said, every study has its limitations. Same thing applies to in vivo studies. So I really want to emphasize that with in vivo studies, we're talking generally about small sample sizes. When you talk about non-human primates, especially, they're expensive to take care of for long periods of time, to feed, etc. And so we're talking about really kind of dozens of animals. They also, because they take so long, that's another limitation in that you can't be constantly doing these on an annual basis and getting new results. It's also important to note that this prion interaction is taking place in an animal, not a human. And so, of course, all the complexities of the human body can't be accounted for. I know a lot of people like to assume, non-human primates, similar to humans, very accurate representation, but it's not really known if that's the case. Both of these non-human primates share some genetic similarity with humans, but there's enough that we can't really be sure how realistic they are. We do have evidence that just 1 or 2 amino acid differences in the PRNP gene, which forms the prion protein, just 1 to 2 differences in that gene can actually play a large role in whether or not conversion can occur. And so, again, just don't take these results and run with them and say it's definitive proof. It's just more evidence, more science to really kind of make educated guesses and to really try to evaluate risk without, of course, seeing whether or not infection can take place in a human, which is very unethical. Also, I want to note a limitation of these in vivo studies is that they really only account for a limited number of CWD prion strains. And so, looking at study results from some of these in vivo studies and making broad conclusions, I would recommend against that, just because all strains haven't been represented. And again, these different strains can have different host ranges. And so you need to take that into consideration when looking at these results. And so just backing up again to a broad level perspective of CWD and public health, which is really kind of what I'm going to school for and it's what I'm doing my project on. Again, want to make it clear that there is no evidence that CWD has infected humans. If you listen to what the experts say, really the consensus on the science completed to date says that the risk is currently presumed to be low, but it is not zero. Of course, consequences of transmission to humans would be severe. And so I'll go with the classic public health byline and say the precautionary principle is warranted in this case. We should not be waiting for definitive proof to take precaution. As a hypothetical situation, if infection in humans were to occur, by the time we notice that happening, with years to decades long incubation periods, putting a stop to exposure wouldn't stop human infections for quite some time. And so again, I'm suggesting we take precautions when possible, make informed decisions, and really that's what I try to emphasize when I talk to people about this issue. And so what can be done? Of course, the common sense approach is to follow the CDC guidelines and again, use precaution whenever possible. So that includes wearing rubber gloves when field dressing your animal, having it tested if you hunt in an area with CWD. If you get it commercially processed, asking them to process it individually to avoid mixing meat. And then, of course, if it does test positive, I would recommend, and the CDC would recommend, against consuming the meat from that animal. And if you want to learn more about CWD and the latest science as far as how you could process the animal, how you could decontaminate your equipment used to process it, I would recommend checking out both our CIDRAP CWD website and the Hunter's Toolkit through the University of Minnesota there. And then I think there's actually more that we can do, and I'll just make a couple more suggestions and plug this paper that I actually helped co-author back in 2019. Some other notable co-authors include Mark Zabel, who's actually the Associate Director of the Prion Research Center at Colorado State, and Brian Appleby is the Director of the National Prion Disease Pathology Surveillance Center, which is the lab at Case Western Reserve University investigating all instances of human prion disease. And so in this paper, we had a section where we really had a call to action. And in that call to action, we identified kind of three primary actions that we would recommend. The first would be expanded investment in CWD research, management, and surveillance efforts. And we noted in the paper that back in 2001, CWD was actually declared a national emergency, probably because BSE was fresh on people's minds. And so federal support was actually expanded for this when it comes to federal financial support, that is. And so, you know, upwards of $10 million plus more was allocated to CWD research every year, to management every year. But since 2011, that money has really been kind of modest and has remained around the $2 million mark. And of course, that has to be split amongst all states or states dealing with CWD, and it has to be split amongst wild and captive or farmed cervid. So you can really see how that money disappears quickly. You know, if you listen to Peter's previous presentations and the work that their group is doing, there are some kind of game-changing steps that could be taken and game-changing developments when we're talking about, you know, potential new diagnostics. We're talking about, you know, maybe a vaccine whenever that would come around. Of course, all of these things can't be discovered unless it has adequate funding. And so we really recommended that be done. Next, we recommended enhanced CWD testing for cervids and really emphasize mandatory testing in areas with CWD detections. I'm actually from Wisconsin myself and hunt in Wisconsin. Wisconsin has some of the highest levels of CWD in the country, and almost all of their testing is still voluntary. So of course, it's really hard for wildlife agency officials to determine where the disease is and at what levels without consistent surveillance taking place. And of course, without mandatory testing, or if hunters choose not to get their animal tested, they don't know if it has CWD. And so you're really talking about exposure to CWD increasing. And again, you're kind of taking away the opportunity to make an informed choice or to avoid exposing yourself to CWD prions if you didn't want to. And then last, we really recommended improving CWD management in cervids. Of course, this is a lot easier said than done, but we recommended looking at AFWA, so the Association of Fish and Wildlife Agencies, developed a best management practices document, an extremely comprehensive, helpful document back in 2018. And they actually identify different policy options that states could take. And so we recommended that states look through those, see what they could implement, and do so just to help prevent and limit the spread of CWD. As I mentioned, when it comes to wildlife management, again, disclaimer, I am not a wildlife manager, but you know, it goes beyond just understanding cervid behavior. I know enough about it to say that. Hunters obviously play a critical role in CWD management. Their buy-in is essential. There's plenty of examples where hunters did not buy into the plan and it kind of backfired on agencies. And so with that in mind, wildlife agencies really spend a lot of time working to understand the attitudes, perceptions, and behaviors of hunters to shape their management plans. And I'm sure you'll get to hear about this more next week with the Minnesota DNR officials who will be presenting on CWD in Minnesota specifically. And of course, the best thing to do is to defer to the experts. And so if you're interested in human dimensions and human dimensions work surrounding CWD, our group hosted a webinar with Vic Adamovich of Alberta, who does work in this area. And so if you want to check that out, again, it's about an hour long and it's available on our CIDRAP CWD YouTube channel. Just if you want, if you're so excited for next week's presentation and want kind of a sneak peek at human dimensions before that, I would recommend checking that out. With that, that's all I got for now. I'm happy to take any questions. And like I said, I appreciate the opportunity to present tonight. So thank you.
Thanks, Corey. That's a really great overview of CWD and humans. It's a really difficult subject, and I think you pointed out well that we're still learning a lot as a research community. I'll start with an excellent question about strains, and I think we'll see how far you want to get into the weeds of this, but I think it really is a good question about what we know about strains. It states what's the distribution of different strains, and can the source of CWD infectivity be linked to the strains? Do we have the same strain in Minnesota and Wisconsin as Colorado? So what really, I think the question is, what do we know about strain distribution?
That's a great question. And like I said, before I kind of get caught up in this, I would recommend checking out the webinar that we did with Debbie McKenzie. She actually talks about this. And so again, another disclaimer, can't have enough. A lot of this strain work is relatively recent. So we're talking within the past decade where researchers even identified multiple strains of CWD. They'll still tell you that it's not known how many strains of CWD exist. Debbie's group actually has identified 4 different strains. And their hypothesis, at least out of Alberta in the presentation that she shared, is that these different foci of disease, so, you talk about Colorado, you talk about Wisconsin, even CWD in Norway, these are kind of speculated to be different strains just based on the species that their CWD is infecting, the genetics of the animals in that population, et cetera. And so I don't know if that covers all of the question. That might be more of a teaser. But yeah, I mean, when you get into genetics and what genotype that a cervid has and what that means for strain development, there has been a lot of work, and Peter could probably tell you more, in looking at the PRNP gene of cervids in different areas and seeing, the proportion of the population that has genes that could essentially create different strains or promulgate different strains there.
Peter, did you want to add anything to what Corey said?
I think Corey did an excellent job. I think just looking at Minnesota alone, the deer in the white-tailed deer in the southern part of the state, they're a little different than the white-tailed deer up in the northern part of the state. And so that geographic variation within species, that's something that we know a lot about. And with what Corey was saying about how strain variation is so recent, he's absolutely spot on. That's correct. And we don't have a good understanding yet of strain variation across different species of cervids because the methods that we have access to are just so coarse. And now that we're getting into these newer technologies, I think we're going to start discovering more and more strains. But just getting back to Corey's point, I think the genetics of that host animal, the geographic variation definitely plays a role into those strains.
There's another question kind of along the same lines. I guess it's a comment more than a question, but maybe something to discuss. The comment is about a study that showed subtype of prion causing scrapie is structurally indistinguishable from the human prion found in those dying of sporadic CJD. And I think that points to the structure of infectious prions in general. And maybe, I think Peter touched on this a little bit in the first webinar, but how does the structure the way we know and can understand and see the structure of infectious prions play into this.
Yeah, I mean, that's a great question. It kind of goes beyond my area of expertise, I would say. But as far as kind of backing up, I'm not sure if they're implying that sporadic CJD could be caused by scrapie, or if they're just saying there's kind of structural similarities. You know, if there's implication there, then of course, when it comes to sporadic CJD, there's really a lot of unknowns. It's sporadic because they basically do not understand where that initial misfolding takes place in humans. Of course, that is the most common human prion disease that we know of. When it comes to the genetics and what that leads to as far as structure goes, again, Peter might be able to offer up a better explanation. And I know just listening back to Debbie's presentation, she touches on that, but basically slight genetic changes that take place in the PRMP gene can lead to slightly different conformations or shapes of prions, and those define these strains. And again, that's really what's thought to be the reason that they might be capable of infecting different species, because it interacts with the normal prions differently than maybe a different shape. Different shaped prion, if you will. And so again, the mechanisms behind that are really unknown. This is kind of a new and emerging area. And I, of course, am not an expert in, you know, that in biochemical science, et cetera. And so again, I'll defer to the experts on that. But of course, these are great questions. So keep them coming. I don't know if I'll be able to answer.
Well, you're doing a great job. And the reality with the structural aspects of these misfolded prion proteins, of like the CWD causing protein, we're still trying to get an accurate structural view of that molecule. Like, again, like the technology has just been lagging behind to do that. So we're going to learn a lot in the next 5 years, I would say, about different strain variants.
Peter, stay on. You might be able to answer this next question too. It talks about diagnostic tests, and I'll throw it out to both of you. But question asks, do the diagnostic tests that we have available detect all of the different strains?
Corey, why don't you take a stab first?
So I can take a stab with, again, a limited understanding only from what I've gathered talking to very smart people working on this. So I know when we talk about prion strains, I can get into kind of what defines them or how they've been really detected at this point. And so I actually made a note to write this down. But the properties of prion strains are defined by stable characteristics, and what those characteristics are: one, these strains might have very different incubation periods. They could cause different clinical symptoms. They might have different neuropathology, so where the prions actually show up in the brain. Then they have different biochemical properties in some instances and different host ranges. So this one really gets into kind of biochemical properties. And there is work they can do using, Western blots, et cetera, where they can see different profiles of the protein. And in some instances, these strains will show up and kind of be basically visible as very different in their protein profiles. From what I'm aware, that's not all of the strains, so you can't, define it just based on that, but that is an indication in some instances that you're dealing with different strains.
Yeah, and with the Western blots, like they're the strains that they pick out, the way that they're differentiating the strains is just a little smudge on a gel, right? So it's a really coarse picture of what's going on. And with the diagnostic tests, are they able to pick up all the strains? This is a really good question, because it turns out that ELISA and immunohistochemistry, I touched on this last week. What they do is they use an enzyme that breaks down protein. It dissolves it. It's called proteinase K, and it breaks everything down. And whatever survives, the prions that survive that are presumably the infectious form. Okay, so you have strains that survive that. The question is, are there certain strains that cause CWD that are susceptible to that enzyme that breaks it down? And if yes, then you're going to miss it, right? They'll go away. And so there could be that there's certain strains that we don't have a good understanding of yet, simply by the process that's being used in these diagnostic tests. And so it's an open question. And this is part of the reason why it's so important to develop diagnostic tests that do not depend on that same methodology.
Peter, that leads into the next question. We're going down the strain trail, which I understand why, because I think strains are really important to prion diseases, and we don't know a lot about strains. So the question talks about differentiating strains. Can we use MALDI-TOF? And MALDI-TOF is, for those who don't know it, it's a way of identifying organisms like bacteria through light scattering patterns. So this gets into some of the advanced diagnostic potential that Peter touched on a little bit last week. But do you want to touch that?
I don't know how much I can touch it. I would say that yes, that's a great question. It's a great question, and without giving too much away of what our lab is working on and what we're working on at the University of Minnesota, if you look at the literature on this, there are papers published where they use light scattering to identify prions. Now, how sensitive that is to look at strain variants, I don't know. It might be too coarse, but in the literature, it definitely shows that those light scattering techniques have potential, right? And I think that's been shown for Scrapie, BSE, and I think CJD. So that's a promising avenue of research, is leveraging methods like MALDI-TOF to detect those misfolded prions. Again, like, it's just debatable about how sensitive that is with respect to different strains. The different strains, remember, it gets back to that three-dimensional conformation, and if there may be slightly different conformations, shapes that would constitute different strains, but what techniques do you have that really do a good job of characterizing that? And right now they're really limited.
Yeah, I think that speaks to what you said earlier, Peter, about the fact that we don't have a good way of identifying the molecular structure of misfolded prions right now. So that, as that advances, I think the diagnostics to identify those will advance also. Well, with that wraps up our questions for tonight. We're nearing the top of the hour, and I want to thank Corey for presenting tonight. Did a great job of giving an overview on a very hard and I think young scientific topic of prion diseases in general, but chronic wasting disease and the human factor of that. Look forward to connecting with everybody next week. And next week, change of pace. We're going to be on Tuesday evening from 6 to 7 p.m. and we'll have a couple of people from the Minnesota DNR with us and talking about CWD in Minnesota and what the DNR has been doing over time and what they're doing this year. So with that, have a good evening, everybody.
Great job, Corey. It was awesome.
Thank you.
Bye, y'all.